And this is where we left off on the last one, just referring to the fact that this making of a protein is very time consuming, very energy consuming, but can be done very quickly because it can be done at multiple sites with multiple RNA. The thing I was saying is once it's released into the cytosol as a polypeptide chain, that chain is modified, it can be modified, and this image that you see here clearly comes from a company called Rockland. But one of the reasons why I put it here is to have you think of, and this isn't something that you're going to be tested on, but have you think of all these different ways that that polypeptide then can be altered and then changed because the goal is to turn it into a functional protein. Then the polypeptide chains can be modified, and during the synthesis of the polypeptide chain, it's going to during the synthesis and definitely afterward, it's going to coil and fold and coil into a three-dimensional shape. But it also may require some modifications before it actually becomes that functional protein. Those modifications will happen in the endoplasmic reticulum, which we have an image of it coming up. I've put this in just as a nice template of that you actually want to make up for yourself. The type of RNA, because we've talked about numerous different types of RNA in the course of teaching. We talked about messenger. That's the one that goes from the DNA to the ribosome. Transfer, that's the translator for protein synthesis, translate what messenger RNA came out into amino acids. Ribosomal RNA, which acts as the catalytic property, that transcript is the precursor to messenger RNA before being processed with some introns in it. That initial messenger RNA that was made, that's not the one that then gets released. You have some splicing going on. And then the small nuclear RNA is also known as the spliceosome. So we have, make yourself a list maybe and understand what each of them does, particularly for the testing situation. Just a reminder, you have two types of ribosomes. You have free ribosomes, and then you have ribosomes that are attached to the endoplasmic reticulum. Free ribosomes, as I said, mostly synthesize proteins that are used in the cytosol. Not that they're simpler or anything like that, but they're just the ones that are right there. You also have membrane-bound ribosomes that are going to make proteins that are going to make their way. You have ribosomes attached to the nuclear envelope, then a vesicle from the nuclear envelope to the endoplasmic reticulum, and then from the endoplasmic reticulum to the Golgi apparatus to outside of the cell. So that's where that endomembrane system. They're going to make their way through that. And then we finally get to the endoplasmic reticulum, and there is another little piece here. If a protein is being made and there's a signal recognition particle, so you have step one, you have the synthesis of the polypeptide, and then what happens, it gets to this this signal recognition particle, this SRP, binds to the polypeptide. And of course, in your textbook and all the way through this whole class, we've talked about induction factors. We've talking about factors with regard to this or that. That means that the process is a little bit more complex than this, but we don't want to throw too many chemicals at you all at once. So there is factors that come in and create this signal. And when that signal is recognized, the signal binds to a receptor on the endoplasmic reticulum, so that SRP binds to a receptor on the endoplasmic reticulum. That endoplasmic reticulum then detaches the polypeptide, and then it is altered, it's resumed, I guess you might say, the synthesis is resumed in the endoplasmic reticulum. That folding and adding and changing. And then that's going to be changed in the endoplasmic reticulum, maybe cut some more, maybe add some more things added. You might have a couple of protein polypeptide chains added together. And that's going to make its way through the endoplasmic reticulum, end up in a vesicle. That vesicle then is going to be sent to the Golgi apparatus and then outside the cell. There are mutations, which we knew we were going to get to mutations, and there are various different types of mutations. Some are much more important than others in terms of changing phenotypes. So you have point mutations. I'm sorry about that, you guys. You have point mutations. They're chemical changes that are just one base pair, and that can be catastrophic in the case of sickle cell anemia, and that can lead to a abnormal protein, which that's what happens in sickle cell anemia. And then you also have point mutations. They can be divided into a couple of categories. That can be substitution, nucleotide pair substitution, or of one or more nucleotide pair insertions or deletions. And the substitution, one nucleotide is replaced with another. Now, of substitutions, they can be silent, which means the substitution, as you can see in this image, in a silent one, we're substituting an A for a G, we still end up with lysine as the protein. So it's never really known that that is a mutation because it still makes the same protein. It can be missense, and what that missense does, it recognizes it. It's a codon that recognizes it as for an amino acid, so it recognizes it and goes forward, but it just doesn't make the correct amino acid, so it's not making the correct one. And then you have nonsense, which unfortunately these might be considered the most severe, because what will happen is it will just lead to a stop codon. And that will then either not create a protein or not create a functional protein. So these are three types of substitution mutations. And then you have these mutations where you have insertion or deletion, and we call these frame shift causing extensive problems. Insertions and deletions are additions or losses of nucleotide pairs in a gene, and these tend to be very disastrous because they don't result in a protein that is useful, or maybe no protein at all. They will change the reading frame of the sequence of messenger RNA, so they will shift the reading frame with regard to the protein that is made. They then lead to serious problems in the individual or wherever this is showing up. Mutations can be spontaneous, but not too many. You have, we talked about maybe 1 in a billion, or there are mutagens. So they are chemicals that are found in the environment, or chemicals that are ingested that cause mutations. And we're talking about things like UV light. We're talking about things like cigarette smoking, etc., that then can cause these mutations to take place. Not all the time, but lead to a higher percentage of chance for mutations. In terms of archaea and bacteria and eukaryotic, compare and contrast here. With regard to archaea, they're prokaryotes, but they share many features of gene expression with eukaryotes. Bacteria and eukaryotes differ in their RNA polymerase. Definitely their termination. Remember the bacteria, it all happens, no nucleus, it all can all happen at once. Archaea tend to resemble eukaryotes in the aspect with regard to the replication of or the aspect of RNA polymerase. Archaea use a complex transcription factors, but their initiation of translation is very similar to bacteria. But so you do have some similarities between archaea and bacteria, and some similarities between archaea and eukaryotes. Bacteria in eukaryotes, transcription and translation are separated. In archaea, transcription and translation are coupled, so it's very similar to bacteria. So you have basically, you can think of archaea, which has always been in all of our studies, is kind of the bridge between bacteria and eukaryotic cells. It's its own category there. This chart doesn't come from your current textbook. It is from an older textbook, but it's a nice chart, and you have it here in terms of compare and contrast the different processes between the different domains. And then one last idea, just to recap what you've done over the past several chapters, is first you started with Mendel not knowing anything about genes or chromosomes or traits, etc. Mendel first thought that there was something there, and then you have this idea of what is a gene, what is the inheritable part of it, and what is a nucleotide base. And then you've moved into understanding what the base is and what the sequence is. So this unit of inheritance, the region of the specific nucleotide sequence in a chromosome, and then how that sequence actually works to make a functional protein or a polypeptide chain. So you've kind of made that journey through understanding what a gene is and what a gene does. We will move on and learn to understand other things as we go forward with the class.